Now today we are going to start with the phylum Porifera. This phylum includes those types of animals which are pore bearing, which have pores on their bodies. Pore bearing animals. That is why they are known as Porifera. Pore bearing animals. So these types of organisms have minute pores in their body for the purpose of ingestion as well as different types of functions. So these organisms have minute pores on their body. That is why they are known as pore bearing animals. So that is why these have the characteristic feature of canal system. Canal system. The characteristic feature is canal system. Now we are going to discuss the important characteristics of phylum Porifera, that is the animals having pores on their bodies. So important characteristics of phylum Porifera. So first characteristic, that all the members are aquatic. That means they may be freshwater or they may be marine. They may be freshwater or they may be marine. So most of the animals of this group are found in marine water, but some of them are found in fresh water also. They can be in the fresh water. So now, as far as the coelom is concerned, these are acoelomate. Acoelomate organisms. What do you mean by acoelomate organisms? These organisms do not possess coelom. Coelom is absent. That is, coelom is the space between gut and body wall. So there is no space between the gut and the body wall, or rather there is no structure like gut. That is why there is no space. So these are termed as acoelomate organisms. Then next characteristic is they are diploblastic animals. They are diploblastic. What do you mean by diploblastic animal? That is, these organisms have only two layers. That is made up of only ectoderm and endoderm. Ectoderm and endoderm. These ectoderm is also known as pinacoderm, and this is also known as gastroderm or coanoderm. So these are diploblastic, means these have only the body of the organism is made up of only ectoderm, which forms the outer layer, and inner layer is formed by the endoderm, gastroderm or the coanoderm. Why the endoderm is known as coanoderm? Because these have flagellated cells known as choanocytes. These choanocytes are flagellated. Just we are going to have a look on the structure, a general structure. These are porocytes. These are porocytes. Porocytes. This is the base part. This is ectoderm, this is endoderm, this is porocyte, this is porocyte, and this is known as ostia. This is known as ostia. The substance which is filled between the ectoderm and endoderm is known as mesoglea. That is known as mesoglea. The ectoderm and the mesoderm comprising of the body of the organization in which mesoglea is present. This is the hair-like structure and this is called osculum. And this cavity is known as spongiocele. This cavity is known as spongiocele. So, and these are called spicules. These are spicules. That is as a skeleton of the organism. That is called spicules. The spicules, the spongiocele, osculum, ectoderm, porocyte, endoderm, mesoglea and the ostia. So this is the basic structure of a porifera that are also known as sponges. These are also known as sponges. So it is evident from this structure, just an introductory structure, that it is having diploblastic structure, ectoderm, endoderm, porocyte, mesoglea, ostia, spongiocele and the spicules. But we have already told you that the endoderm is also known as choanoderm. Why? Because choanocytes, specialized choanocytes are present in the endoderm. These are flagellated cells. These are flagellated cells. These are flagellated cells. These are flagellated cells forming the choanoderm or the endoderm. So this is the overall structure of the introductory structure of a porifera, that is pore bearing animals. So pore bearing animals are diploblastic, having ectoderm and endoderm. Endoderm is also known as choanoderm because it is having choanocytes. These are specialized cells which are found only in poriferans. Now these are acoelomate. Why? Because these are acoelomate, because no coelom is present. Now as far as the canal system is concerned, these are having the characteristic feature of canal system. That is, through this system it performs its respiration as well as feeding. Now canal system is the characteristic feature of poriferans. These are of three types. Number one, ascon type. Number two, cycon type. And number three, that is leucon type. Ascon type, cycon type and the leucon type. So now we are going to discuss with the ascon type. And ascon type, it is the simplest type, and water comes through the ostia into the spongiocele and then it comes out through osculum. But in cycon type, water, then ostia, then prosopile, then apopyle, and then to osculum. And in Leucon type, we have the water, then to ostia, then in current canal, then prosopile, then apopyle, and then X current canal, and then osculum. So osculum is acting as anus and ostia is considered as mouth of the organism. So these are the basic three types: Ascon, Sycon and Leucon type. In Ascon type, it is the simplest type. Water enters through the ostia, goes into the spongocoel and then out through the osculum. Then through Sycon, water, ostia, prosopile, apopyle and osculum. Leucon, from water to ostia, incurrent canal, then prosopile, apopyle, excurrent canal and the osculum. The material is excluded out or it is thrown out through the osculum. Now coming on this is the characteristic feature of phylum Porifera. Now coming on the next characteristic, as far as the nutrition is concerned, that is how it performs its nutrition. It is holozoic type. Holozoic type means it engulfs whole organism, especially water comes. Say this is the organism outline structure. This one. Water comes through the ostia and with the along with the water, the organisms or the planktons comes inside the spongocoel. This is the spongocoel. And after digestion through osculum, material is thrown out. So it is basically a holozoic nutrition, but it is a planktonic feeder. It is a planktonic feeder. So water comes through the ostia, it will be digested in the food vacuole inside the spongocoel, and the excluded out or the excretory material is thrown out with the help of osculum. Now excretion. So if it performs nutrition, it should excrete. The excretion is ammonotelic. That is, it performs or it excretes ammonia. So excretion is ammonotelic, that is, it excretes ammonia because they are mostly or completely aquatic. Now respiration is aerobic. That is, it performs its respiration in the presence of oxygen and it will be absent. If oxygen is absent, there will be no existence of this poriferans. So excretion is ammonotelic, nutrition is holozoic planktonic feeder, and respiration is aerobic. And as far as its number is concerned, that is reproduction is concerned, it can be by asexual method, that is by budding, reduction bodies, that is with the help of fragmentation, and each fragment can form the new individual. And sexual reproduction, that is by the production of male and female gametes. The sexual reproduction is cross and internal. That is, the zygote develops in the mesenchyme or the mesoglea. So these are the basic characteristics of phylum Porifera. So once again we have to see the characteristic, that is they are diploblastic. Then we have what? These are having the two layers, ectoderm and endoderm, and the middle there is no layer. This is non-cellular layer known as mesoglea. This is acoelomate, that is no coelom. Third thing is that nutrition is holozoic. Fourth is excretion is ammonotelic, that is it excrete ammonia. Then we have its reproduction by sexual, asexual. Sexually by the production of zygote, reduction bodies, and asexually by budding. So these are the basic characteristics of phylum Porifera. Now this poriferans can be categorized, that is classification. The basis of classification. So every group is characterized on the basis of some specific character, that is called spicules. On the basis of type of material which is made from, the spicules are made up of, that are considered into three categories, that is calcarea, then hexactinellida, and demospongia. Calcarea, hexactinellida, and demospongia. Calcarea or calcispongia. Calcarea or calcispongia. Spicules are made up of calcium carbonate. So in this group, calcarea, the calcarea or calcispongia, spicules are made up of calcium carbonate. In this, spicules are made up of silica. And in demospongia, spicules are made up of spongin fibers. Calcarea, hexactinellida, and demospongia. Now we are going to discuss about a member of this phylum Porifera. Now this Porifera, Leucosolenia. Now this is a sessile colonial and marine sponge. Sessile means it is do not having the property to move. It cannot locomote or it cannot change its position. Colonial, that they are not solitary, they are found in the form of groups. And marine, that is they are always found in the marine water, that is sea water or ocean water. So, and these possess radial symmetry. That is, from any plane it can be divided into two equal halves. Then on the basis of nutrition, the nutrition, as far as nutrition is concerned, the Leucosolenia performs Holozoic type of nutrition and plankton feeder. That is, with the help of water current, planktons enters the spongocoel, and with the help of food vacuole, they engulf the feeders, the planktons, and the undigested food is excreted out through the osculum. So, food vacuole helps in digestion. So, digestion is intracellular. It is intracellular. Now, the medium or the pH of the food vacuole. pH of the food vacuole is first acidic, then alkaline. As in higher animals, the medium of the stomach is acidic, and then the alkaline nature of the succus entericus or the intestinal fluid. So it becomes first acidic, then alkaline for the proper digestion of each and every component of the food. So this was the nutrition of the Leucosolenia. So as we have seen that this is a diploblastic animal. So it is having two types of layers, ectoderm and mesoderm. Ectoderm, that is pinacoderm, and endoderm, that is coanoderm. And in the mesenchyme, the non-cellular layer present in between the ectoderm and the endoderm is known as mesenchyme or mesoglea mesenchyme or mesoglea. Now this mesoglea is non-cellular. It is non-cellular. So let's have a look on the mesenchyme or the mesoglea. So mesoglea, it is non-cellular part. It is non-cellular part of the organism having spicules and amoebocytes. That is having different types of cells such as germ cells. Then we have archaeocytes. Then we have thesiocytes. Then we have scleroblast cells. So these are the different main types of cells which is present in the non-cellular part, and the development of the zygote takes place in the mesoglea, not in the spongocoel. Now we have seen the nutrition of the organism, then we have the respiration as well as excretion. Respiration is aerobic and excretion is amonotelic. Amonotelic means that it produces ammonia, and this is performing aerobic respiration, and both the function is performed by general body surface, because there is no specialized organ found for these purpose. General body surface. The general body surface amonotelic, excretion is amonotelic, respiration is aerobic by general body surface. So now, as far as the reproduction is concerned, that is how it increases its number. So we are going to have the look on the increase in number of the poriferans. Now, reproduction of leucosolenia. So it increases its number with the help of budding asexually and by the help of fragmentation and by the production of ova and sperm cells by sexual reproduction. And the fertilization is cross and internal, that is, it takes place inside the body, not outside the body. So it is cross and internal. So the development is indirect. As far as development is concerned, that is development of the zygote into the adult form, is indirect because there is the presence of a larva called amphiblastula. The amphiblastula larva is found in the life cycle of the leucosolenia. That is why the development is called indirect type of development. That is having a larva called amphiblastula. It is a solid larva. Now, a very interesting phenomena shown by these poriferans is regeneration. What do you mean by regeneration? That is forming of its own body from the differentiated cells. So this phenomena was discovered by H.V. Wilson. That is, if you chop all the different types of porifera, all the different types of sponges, and mix them, and when you reproduce or when you flow in the water, then different types of cells will rearrange to form peculiar type of sponges, not mixed type of sponges. So this type of regeneration is found in poriferans or the sponges. So this was shown by H.V. Wilson. So these are the poriferans. Once again we have to rewind that poriferans are the organisms which are bearing the pores on their bodies, and through these pores they ingest food through the water current and digest it inside the spongiocele, and through the osculum they will excrete the undigested food. So these are all about the poriferans. Now, next group of organism which is slightly more advanced than poriferans are coelentrates, or the phylum Coelentrata or Cnidaria. That is a body cavity, a characteristic body cavity is found in the Coelentrata, that is known as coelentron. The body cavity is known as coelentron. That is why these are known as Coelentrata or the Cnidaria. Now, as we have seen, the poriferans have the characteristic feature of canal system. These are having the coelentron. Now, the characteristic feature. Characteristic features. So, number one, as we have seen in the poriferan, these are also diploblastic animals. Diploblastic animals. What do you mean by diploblastic animals? These are having ectoderm and endoderm. But these do not possess choanocytes. Choanocytes are found only in poriferans or sponges. And the middle layer, that is non-cellular, known as mesoglea. Third thing, that is they are also acoelomate. That is, they do not possess coelom, or coelom is absent. That is, coelom is absent in this case. So diploblastic, mesoglea is present in between ectoderm and endoderm, and acoelomate, that is coelom is absent. But these organisms have body cavity called coelentron. The body cavity is known as coelentron. Now, these organisms are found in more than one form. More than one form means what? They are found in cylindrical structure and umbrella-like structure. Cylindrical structure is known as polyp. The cylindrical structure is known as polyp, and the umbrella shape is known as medusa. Polyp and medusa. Cylindrical form is known as polyp, and umbrella like structure is known as medusa. The polyp is asexual part and medusa is sexual part. That is, gonads are found only in medusa part and not in polyp part. So we can say that there is alternation of generation. Alternation of generation takes place, but it is a specialized type of alternation of generation where there is no alternation of haploid or diploid, but there is the change in the structure, asexual and sexual part. So that is known as metagenesis. This part is known as metagenesis. So alternation, it is not true alternation but called metagenesis. Appearance of cylindrical as well as umbrella like structure. So when we characterize it or differentiate into the categories, they can be into three parts: hydrozoa, scyphozoa and anthozoa. hydrozoa, scyphozoa and anthozoa. Hydrozoa includes those organisms which have polyp and medusa. Alternation of polyp and medusa. Scyphozoa only medusa present, polyp absent. Polyp present, medusa absent. So this includes corals. This includes jellyfishes, Aurelia, and this includes Hydra, Obelia. Hydrozoa, Scyphozoa and Anthozoa. But Hydra do not have the medusa form, but other characteristics are same. That is why they are under the category Hydrozoa. Hydrozoa, Scyphozoa and Anthozoa. Thus the phylum Coelentrates are also diploblastic, that it is also having the ectoderm and the endoderm. These are also acoelomate, that is coelom is absent. There is no structure like the gut, so there is no space between the gut and there is no question of space between the gut and the wall, body wall. Now as far as the categorization is present, these are categorized on the basis of the structure present, that is called polyp and medusa. Two types of structures are present, polyp and medusa. Polyp and medusa is found in hydrozoa, only polyp is present in anthozoa and only medusa is present in scyphozoa. In scyphozoa, polyp stage is absent, in anthozoa, medusa stage is absent. So where there is alternation of the polyp and the medusa structure takes place, that is known as metagenesis. So it is basically found in Obelia. Metagenesis. This is a specialized phenomena found in Obelia. So hydrozoa, scyphozoa and the anthozoa. Now as we have seen Locomotion in the Porifera, we have to explain the Nidaria with the help of an example, that is Hydra. Hydra. Hydra is very minute, but this is coming under the Coelentrata because it has the coelentron. So let's have a outline structure like this. These have tentacles like this. This is pedal disc. This is gastric portion. This is pedal portion. These are hypostome. This is hypostome. This is mouth, and these are tentacles. These are tentacles. So the hydra looks like this. So it is also known as freshwater polyp. It has many species, but Hydra viridissima is green in color due to the presence of algae. Hydra viridissima. It is green in color because of the presence of algae. So it is also known as freshwater polyp belonging to the group Hydrozoa. So this is the basic structure: the mouth, hypostome, gastric portion, pedal portion and pedal disc. Now, as you can see that there is only one opening for the ingestion as well as for the egestion, that is excretion. So this type of body organization is known as blind sac body plan. This is known as blind sac body plan. Why this is called blind sac body plan? Because there is only one opening. Anus is absent. The ingestion as well as egestion of the material or the food particles takes place through the one opening, that is mouth, and that is also acting as anus. That is why this is known as blind sac body plan. Now Hydra is diploblastic. Hydra is diploblastic organism, that is it is having ectoderm as well as endoderm, and between the ecto and the endoderm mesoglea is present. Mesoglea is non-cellular jelly-like substance that is present between the ectoderm and the endoderm. Now, the cavity, this cavity is known as coelentron. So, as far as its nutrition is concerned, it is carnivorous. It is very active during the capturing of the prey, and its favorite food is cyclops and daphnia. Cyclops and daphnia, these are their favorite food, and these organisms have special types of organs or the cells for capturing the prey and for the movement, known as cnidoblasts, or stinging cells. These are known as stinging cells or cnidoblasts. So these cnidoblasts are the characteristic of the phylum Coelentrata as well as for the Hydra. So these are of four types: penetrant, volvent, large glutinant and small glutinant. Large glutinant and small glutinant. These help in capture of prey. Helps in capture of prey. But there is no hypnotoxin. No hypnotoxin in large glutinant. Hypnotoxins are found in penetrants and volvents. The cnidocyl ejects out and it will enter the prey and causes their death due to hypnotoxin. But large glutinant also helps in the capture of prey, but there is no hypnotoxin present in the large glutinant. So as far as the small glutinant is concerned, this helps in locomotion. That is, it helps in the attachment of the pedal disc with the substratum. Small glutinate, large glutinate, volvent and penetrants. The toxin through which it causes the death of the prey is hypnotoxin. This comprises of phenols, penetrant, volvent, large glutinate and small glutinate. With the help of these nidoblasts or nidocells or stinging cells, it causes the capture of the prey. Because it is carnivorous and it is an active carnivorous, and the favorite food is cyclops and daphnia. Now, as far as the locomotion is concerned, so as it has the tentacles as well as the nidoblast cells, it can locomote. As far as the locomotion is concerned, it can change its position from one place to the other. So it performs various types of locomotion such as gliding, walking, somersaulting and jumping, as well as swimming. So it can glide, it can leap, it can walk on the substratum, somersaulting, jumping as well as swimming. So it performs different types of locomotion movement, and it can change its position from one place to the other, and it can capture the prey actively. That is why these are known as freshwater polyps. Now, because these have barrel-like structure, polyp structure. Now, as far as respiration is concerned, the respiration is aerobic. That is, it cannot live without oxygen. If oxygen is absent, then this organism can also be depleted. Now, reproduction of the organism, reproduction. It can reproduce asexually by budding, sexually with the help of formation of zygote and fragmentation. Now let's see each one of them. Asexual reproduction by budding. So what helps in budding? What happens in budding? Say this is an